Hi everyone, it's Mr. Sinti, and today I'd like to talk to you about bacteria, and I'd like to talk to you about archaea, and both of those two domains are considered to be prokaryotic organisms. And so I want to give you a little background about prokaryotes today because a lot of molecular biology, molecular genetics works with these prokaryotic organisms. And so I think a little background is necessary, and so I hope you enjoy it. So let's jump right into the conversation. And so one of the first things that I'm going to say about um, prokaryotes is that they're everywhere. And so they're all over us, they're all over the table, they're all over your laptop, they're inside of us. And so it's remarkable the number of organisms we're talking about. And so these are the earliest organisms that we believe evolved on the planet Earth. And so they're the oldest and ancient, and they've been with us, and they're very simple, but it's a lesson in life, and that simple sort of gets the job done just as well as complex does. And so they dominate, and so they dominate the biosphere. There's so many of them. They have the most biomass out of any class in biology, and they inhabit everything. They're in the soil, you're in their mouth, the skin, and so again, you could sort of pull up a little handful of soil from your backyard, and there'd be more prokaryotes there than there were ever people ever alive on the earth. It's remarkable. Here's a little picture. This is an actual photograph of E. coli. It sort of looks like all these little tentacles coming off of it are these structures called pili, and they're hollow, and they help for bacteria to actually exchange DNA with another bacteria, something we'll talk about. I have a little model here. I guess this is Miss E. coli, and you can see here all of her little pili that are coming off right here. It's kind of cute. So, what I want to say about the prokaryotes is that they're found everywhere. When I say everywhere, even in places on the earth where we believed that no living thing can sustain, like deep under glaciers or in the hot springs of Yellowstone National Park, or places that are simply too hot, too salty, too acidic, too alkaline for any eukaryotic organism to live, but yet prokaryotes are able to live there. And you can see these colorful formations here in yellows and reds are an example of prokaryotes living in these pretty brutal conditions. And so one of the things is that there's a big misconception about prokaryotes and that they're evil. And so I'm not going to be Pollyanna about it, because most of them, even though most of them are good, some of them can be horrifically harmful. And so just to point out, during the 14th century, the bubonic plague, which literally killed 25% of the human population, was caused by bacteria. And so we still have some dangerous bacteria with us today, like TB, things like cholera, and bacteria that can cause food poisoning. Maybe you know the name Salmonella, which can be found in chicken, and that can cause problems as well. But the truth is, those are just the ones giving the rest a bad name. Most bacteria are either benign, meaning they're not harmful at all, or could even be beneficial. And so some of these bacteria live in our intestines, and they help actually produce vitamins. And so therefore we don't have to eat those because they're inside making it for us. And so one of the main things, we had talked about this before, is that prokaryotes even recycle the elements. So when something dies, bacteria decompose it, put it into the soil, and make it available for autotrophs. And so they can even take nitrogen in the soil and put it back into the atmosphere. So they recycle nitrogen, they recycle carbon. They're the recyclers. Most important. That alone signifies prokaryotes as being the most important organism on the earth. That statement right there, in and of itself, need not go further. But they also live in close association with plants and animals and fungi. And what's interesting is, I mentioned that they help produce vitamins, but in other cases, when they live with plants, they help to nitrogen fix nitrogen from the air and produce ammonia, which plants can use in the soil. We even believe that eukaryotic organisms, in particular the chloroplast and the mitochondria, were once free-living prokaryotes. And we suspect this because both those two organelles have ribosomes, which are very similar to prokaryotic ribosomes, and they have small circular pieces of DNA that, when sequenced, resemble that of bacteria. So I find that to be kind of interesting, that we're part prokaryote. Even our cells are part prokaryote, even though there's so many living inside of us. Well, in terms of classification, classification is something that's subject to change, and so we're always sorting organisms based on new information, which is fine. Most of this new information is coming out of DNA sequencing, but this is where we are today. Molecular evidence, like sequencing, accumulated over the last 20 or so years, has made biologists believe that there's really two huge branches of prokaryotes. Before, we used to think that there was a single kingdom called Monera, but now we believe that we're going to take it above the kingdom Monera and get rid of that. We're going to say that there's a domain, which is even larger than a kingdom. And so we're going to say that there's a domain called archaea, and these are both archaea and bacteria are prokaryotes, which mean they're single-celled, they're very teeny tiny, and they have no nucleus. And so archaea are the ones, and there are billions of them, they're the ones that live in extreme conditions. These are the ones that are kind of the wild ones, can do high tolerance for temperature, hot, cold. They can also do harsh pH. They can also do salinity. And so they have weird abilities, both biochemical and physiological characteristics that allow that to occur. So when you look at the bigger idea, I'm not going into below this would be below domain eukaryote. Instead of all eukaryotes are the different kingdoms, so the plant kingdom, etc., animal kingdom, protista. Then there's kingdom, phylum, class, order, family, genus, and species, going all the way down. But on this larger level, we like to think that there's three domains. There's the domain bacteria and domain archaea, and both of these are prokaryote. That's what we're talking about. And then you're like, well, why don't we talk about eukaryotic a little bit more? We often do, but this is by far, by really far, the most numerous organisms on the planet Earth. And so what's interesting is we believe that we're more closely related to archaea, as you can see here, the branch off, than we are to bacteria. So archaea are more similar to eukaryotes than they would be outright to bacteria. And so let's take a look here. Whoops, a little too quickly. So in terms of the shapes, this is sort of just a general conversation about prokaryotes, bacteria in particular. What do they look like? What are some of the structures? They look simple. They're single-celled. They could either be circular, broad-shaped, or a spiral, or cocci, if you will, bacillus, and spirilla, right over here. And so they could be helical in shape. And so there's a rather assortment there. Can you imagine all of this all over our body and our body? And so nearly all prokaryotes have a cell wall and a plasma membrane. And of course they have ribosomes as well. And so the cell wall, like in plants, helps them to maintain their shape, and it's a physical protection. It prevents them from bursting in a hypotonic environment. So that's really important to bacteria. So if your goal is to kill a bacteria, maybe one way to do it is to disrupt its cell wall, and then simply water would go in and cause the cell to lysis. And so its cell wall is a little bit different than a plant cell wall. Maybe you can say it's a lot different. You may recall that the plant cell wall is made up of cellulose, but the prokaryotic cell wall is made up of something called peptidoglycan. And this is actually rather important when you consider the number of prokaryotes. So this is an important molecule. And basically, it's sort of like sheets. It's a big polymer, and it's made up of sugars and nitrogen together. Sugars, nitrogen, and some polypeptides. And it's an interesting little structure. sort of like fiberglass, if you will. Although, here's one of the big differences between bacteria and archaea. Archaea do not have this peptidoglycan cell wall. And so, one could say in terms of classifying bacteria, you can classify them a couple of ways, and one of the main ways in which you can classify them is how they stain. And we have these different purple stains and red stains, and one of them is called Gram stain, and it's useful because it picks up this peptidoglycan wall. So when you add these dyes to the bacteria, do you notice here that they're sort of purple in appearance? They're sort of rod-shaped here. The wall, or peptidoglycan wall, which is sheets of sugar and proteins right in here, which is on outside of the cell membrane. So it's cell wall and then cell membrane. The stain picks it up, and so it gives this sort of purple appearance. And so they have this, they're called gram positive if they have a big peptidoglycan layer. Some bacteria do not have that big peptidoglycan layer on the outside. Instead, they have a membrane that has a lot of sugar on the outside of it. Then they have this really thin peptidoglycan and then another cell membrane inside. So these are sort of like peptidoglycan less. And so when you stain them, they don't have that purple appearance, and so they're kind of red in color. So they're called gram negative. As it turns out, these gram negative ones are more so the harmful bacteria. These are the troublemakers, if you will. And we think that it has something to do with the fact that this outer membrane makes them elusive and makes it difficult for the defensive systems of the host. And so I made this red. The gram negative ones are the ones that are more threatening. They're elusive to the host defensive systems, meaning white blood cells. And they're commonly the ones that are resistant, if any, are resistant to antibiotics. And so it's sort of this membrane impedes antibiotics, and so the antibiotics don't hurt them. Normally what happens is when you add an antibiotic such as penicillin, you can see here's the mold growing, penicillium, and then you can see this sort of halo of death. The bacteria die, and it's like, why do they die? Because this chemical penicillin prevents the bacteria from producing a really nice peptidoglycan wall, and without that, they're vulnerable to water, and then water comes into them, and they lysis. And so when we take antibiotics, we need to drink plenty of fluid in order to cause them to burst. And then, can I add one more layer, if you don't mind? Even still, some bacteria have yet a capsule on the outside of their cell wall, and this is a polysaccharide mostly, and it makes it kind of sticky and in some ways slippery. You might recall when we were talking about Fred Griffith's experiments in England and London during the 1940s and 30s. He was working with S bacteria that had this sugary coat on the outside, this capsule. And so oftentimes bacteria that are found on our teeth have this capsule as well, and it helps them to stick to each other, forming colonies. And it also makes them kind of resistant to host defenses. So some have a sugary capsule, some do not. I mentioned before, every living thing has ribosomes. Prokaryotes have one big circular chromosome, and sometimes there's circular pieces called plasmids. These are those pili that are extensions that come out that are hollow. And then bacteria move around by flagella, which is this big protein of microtubules which whips back and forth and helps to propel them. So here's a picture of the pili. I mentioned before, my little puppet here of E. coli, that these little extensions are called pili. And what the pili are used for is that they're hollow, and so inside the cell there's DNA in here. Sometimes DNA can travel can travel through these pili. Let's go a different color here. So the DNA will move through the pili and will actually engage another bacteria. And so if DNA will move from if DNA moves from one organism through the pili to another, that's what we mean by sexual reproduction or conjugation. That's ultimately what we're talking about, is the DNA moves from, from one bacteria to another. Excuse me. So sometimes the pili is actually called a sex pilus. And so this is a kind of an R-rated photograph showing bacteria reproducing with one another. And inside that pilus is DNA moving from one to another. We'll come back to this notion a little bit later when we talk about how genes are transferred between bacteria. Here's a few more pictures of bacterial conjugation. And then finally, many bacteria are mobile. And so they can move around by these flagella, and helps them to be able to transport. You might say to yourself, where are they going? And so if they're mobile, I can't imagine that they need to travel very far. I never really probably considered it. But the truth is, you may recall this kind of a conversation in that bacteria, though simple, these corkscrew ones, spirilla, can actually sort of just rotate around and move around. But if they're in an area where they don't like, they would want to move out of it. Or if they're in an area where they notice that the environment is particularly good, they might want to like sort of swim towards something that's good or swim away from something that's bad. And you may recall that being called taxis. So in other words, if the environment is heterogeneous, some areas are good, some are bad, prokaryotes are capable of this orientated movement. It's certainly not very far, but it's a movement toward or away from a stimulus. So they can have a positive taxis. So positive chemotaxis, meaning they're going toward something good, or negative, moving away. They can even move toward or away from light, or toward or away from a magnetic field. It's kind of impressive. So I hope you enjoyed our little conversation of prokaryotes and the domain of archaea and the domain of bacteria. Thanks for watching.